Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is more of an open format discussion of additional articles, conferences, or major events in the field that we'd like to highlight. My name is Darleen Sandoval, and I'm joined by my podcast co-hosts, Dr. Kirk Habiger and Dr. Kevin Williams. Happy Tuesday. How are you guys doing?
Hey, Dar, happy Tuesday. First day of summer officially, right?
Yeah.
Good to see you all.
Kevin's having a little rougher day today.
Yeah, we had some weather move through. We're dealing with it, but it's brighter skies right now, so.
The sun has come out. All right. Well, let's dig into what we have in store for our listeners today. Kirk, why don't you get us started?
Thanks, Dar. This month, we'll talk to senior author Dr. Ralph DeFronzo about his group's new paper regarding the effect of dapagliflozin on renal and hepatic glucose kinetics in type 2 diabetics and normal glycemic subjects.
And Kevin, what do we have in store for the Sweet Talk segment?
Yeah, today we're highlighting the latest Point-Counterpoint article from the journal, where experts debate viewpoints on hot topics in diabetes. Today we're exploring the regulation of KATP channels in beta cells. We have Dr. Guy Rutter and Dr. Ian Sweet, who support the canonical model, which suggests that mitochondrial oxidative phosphorylation drives KATP channel activity. Dr. Matt Merrins and Dr. Dick Kibbey are challenging this model. They propose that glycolysis and pyruvate kinase are more bioenergetically favorable for regulating KATP channels in beta cells. I encourage all our listeners to stay tuned for a thought-provoking discussion on these competing models and their potential impact on diabetes treatment.
These point-counterpoint segments are going to be really fun moving forward. We have a few more coming up this year. Is that right, Kevin?
That's right. Yeah, looking forward to it.
All right, let's get started.
Today, we're going to discuss effect of dapagliflozin on renal and hepatic glucose kinetics in type 2 diabetes and normal glycemic subjects by Shi Chen and colleagues. We're joined today by the senior author, Dr. Ralph DeFronzo. Dr. DeFronzo is a professor of medicine and chief of the diabetes division at University of Texas Health in San Antonio. Welcome, Dr. DeFronzo. Congratulations on your paper, and thank you for being a part of this month's podcast.
Yeah, it's a great pleasure for me to be with you today. This whole story of SGLT2 inhibitors and their effect on the kidney and the heart and glucose metabolism is a really exciting one. Has lots of important physiological and clinical implications. So I'm delighted to be with you today.
Awesome. We like to start with a fun icebreaker question. And so I thought I would ask you, since you've had a very successful career in science, if you were not in science, what career do you think you would have pursued?
Yeah, so what I would have done is coming out of high school, could have signed a contract to play professional baseball with the Boston Red Sox. So who knows, you may have seen me in Fenway Park hitting balls off the Green Monster.
That's awesome.
That's very cool. All right, before we get started, we'd like to give you the opportunity to highlight any other contributing authors or collaborators to this work, if you have anyone in mind.
Yeah, there are a couple of people who played a really critical, important role in the study. And of course, the first person on the publication, Stephen Chen, he's the person that did all the legwork. So we really need to give a lot of credit to Stephen. Bob Chilton is the cardiologist who did the hepatic renal vein catheterization. So without him, we couldn't have accomplished, I think, these incredibly impressive results. So those are two very special people. And Dr. Sersosimo also played a very important role in carrying out the studies. So it's a great team to work with.
Awesome. Well, Dr. DeFronzo, can you provide a summary of your research program, maybe a 30,000-foot view, and then a bit about how that led to this project?
Yeah, so we've been, I think, very active in defining what's the pathophysiology of type 2 diabetes. And I think everybody is very familiar with my ominous octet Banting lecture at the ADA in 2008, which really has set the stage for our understanding of what causes diabetes. And of course, if we understand what causes diabetes, then we'll understand better what drugs work and what drugs don't work. And that's really been my focus. Now, specifically, I have a little bit of an unusual background because I've done both a nephrology fellowship and an endocrine diabetes fellowship. And during my nephrology fellowship, I was trained as a micropumpist. And I sat there under a microscope and put these little tubules, cannulated these tubules with micropipettes. And I was particularly interested in glucose and phosphate transport in the proximal tubule. And I worked with a drug called Florazin, which comes from the bark of the apple trees when the apple trees bloom in the spring. And Florazin blocks glucose transport. But actually, at that time, I was more interested in phosphate transport. And so, as a side effect, I also learned a lot about glucose transport. And it dawned upon me, hey, why don't we just treat diabetes by blocking glucose transport in the kidney? And that will just let the glucose come out in the urine, the blood sugar level will come down. And it's such a simple concept. No one thought about it, but it's quite ingenious if you think about it. And in fact, it had to work. And of course, we subsequently went on and showed in animal studies that Florazin actually could normalize glucose tolerance in a number of diabetic animals models. And when you corrected glucotoxicity, you improved insulin sensitivity in muscle, and you improved beta cell function. So all of the things that I postulated would happen from nephrology studies actually turned out to be true when we came to our diabetic studies in animals.
Very interesting. So there's been a lot of excitement over these drugs especially in the recent years in their ability to lower HbA1c, but also these effects of lowering body weight and also their cardio and renal protective effects. However, even given this lowering of HbA1c, the body of literature suggests there's a seemingly paradoxical finding or impact of these drugs. So, can you tell us a little bit about that finding and based on the body of work, what you hypothesized was driving that effect?
Sure, love to do it. So when we did these original studies, we pretty much postulated exactly what was going to happen, except for one aspect, the cardiovascular component. I'll come back to that in a second. But it was very clear to us if you block glucose transport in the kidney, glucose should come out in the urine. And to the extent you excreted a certain amount of glucose, you could predict what should be the drop in the blood sugar level. And one of the surprising findings we found out was that the drop in the blood glucose, based upon how much glucose came out in the urine, was only 50% of what we anticipated. So this was a little bit strange, and I'm going to come back to this. But we also postulated at the same time that the mechanisms by which you block the glucose transport in the proximal tubule by delivering more glucose and sodium to the juxtaglomerulosa apparatus actually should correct the pathophysiologic mechanisms that are leading to diabetic kidney disease. So we postulated at that time, not only would this drug be good for lowering your glucose, although not quite as much as we expected, you lose glucose, you lose calories in the urine, you would lose weight, and we would protect your kidneys. All of these postulates came out to be true. What was an enormous surprise to me, and I think everybody in the world, was the incredible effects that these drugs had on the cardiovascular system. No one expected this. And in fact, we can maybe thank Dr. Stephen Nissen, who sort of enticed the Food and Drug Administration to require that for every diabetes drug that's approved, that you have to do a cardiovascular outcome study. Some of us felt that this was a little bit excessive, but obviously Dr. Nissen had great insight. And in carrying out the cardiovascular studies with the SGLT2 inhibitors, we found this incredible protective effect in heart failure and prevention of cardiovascular events. Now, why did we sort of underestimate the potential effect of these drugs in terms of the drop in the A1C? So as we were carrying out the studies using tracers, treated glucose, to follow endogenous glucose production, we found that when the glucose appeared in the urine, literally within 15 to 30 minutes, there was a stimulus that led to an increase in hepatic glucose production. So we're losing glucose in the urine, but now the body itself is now producing glucose. And what we showed is that the production of glucose from within the body offset by about 50% the amount of glucose that was coming out in the urine. So, of course, that now set into play another set of experiments to try to define what was responsible for the increase in endogenous glucose production. Now, there are only two organs within the body that can produce glucose. One is the liver, and that really is the major source, 80 or 90%, maybe even more. The other is the kidney. And there's been a large controversy is what is the kidney's contribution? I'd have to say I've always been more on the side the kidney really is not that much of a contributor, but there are other people who have felt the kidney was producing a significant amount. Now, before we get into the paper and which organ is playing a role here, we know what controls hepatic glucose production is the insulin level, glucagon level, and the plasma glucose concentration itself. So if your insulin level drops, hepatic glucose production goes up. And if your glucagon level goes up, hepatic glucose production goes up. And when I give you one of these SGLT2 inhibitors, the one we're going to talk about later is Dapagliflozin, but basically the results are the same with all of them. As the glucose comes out in the urine and the blood glucose drops, what happens is the insulin level drops. That's going to favor the liver producing glucose. We also noted that the glucagon levels went up, and exactly why that happened still, I would say, is somewhat controversial. So we actually did what we call pancreatic clamps, where we clamped the insulin level, wouldn't let it change. We clamped the glucagon level, wouldn't let it change, and clamped the glucose level. And then we gave the dapagliflozin. And what we showed, we still saw this big increase in endogenous glucose production. So we really ruled out insulin, glucagon, and changes in the plasma glucose concentration. So the next question was, well, why was this happening? We then subsequently went on and did some studies where we, and our postulate was that what was happening is that when the glucose appeared in the urine, we were activating the renal nerves, and the renal nerves were sending a signal to the liver to produce glucose. Such a connection, can you imagine here in 2024, had never been described before.
Yeah.
How could this physiology be unknown? And so we took people who were going to have a renal transplant, but we took people who had polycystic kidney disease. And so when you remove the polycystic kidneys because they're large and obstructive and they be associated with pain and hematuria, then you get the transplanted kidney. So now the transplanted kidney is denervated. There's no way it can connect with anything. And your endogenous kidneys, they've been removed. What we found is that now when we give the dapagliflozin, the increase in endogenous glucose production was blunted. And so that suggested to us that the renal nerves were playing a critically important role here. So then the next thing we wanted to know, of course, and that's what's led up to the current paper that we're talking about, are the kidneys involved intrinsically? Is there something that the dapagliflozin is doing that stimulates the kidneys to make glucose, or is there an unknown signal which is being sent by the kidneys to the liver? So in the paper that we're discussing today, what we did is we put a catheter into the renal vein so that we could directly measure glucose production by the kidney. And we then used tritiated glucose to measure total body glucose production. So the tritiated glucose tells me how much glucose is coming from the liver plus the kidney. From the renal vein catheterization, that tells us how much glucose is coming from the kidney. And the difference between the two is what the liver is producing. And in fact, what the result showed is the kidney basically was not producing any glucose in response to dapagliflozin. All of the increase in glucose production was coming from the liver, which is what we actually thought was going to be the result. But again, it's nice to think and postulate. It's also nice to do the definitive experiment that confirms your postulate.
Yeah, thank you for that.
So you've touched a little bit on the methodology and also on some of your hypotheses as to how this is happening. Is there any room for a direct effect of these SGLT2 inhibitors on the hepatocyte itself? Or do you think that is it's exclusively coming inter-organ communication?
Yeah, I think that one could postulate that there is an off-target effect. There are no SGLT2 inhibitors in the liver. We now know that all of the increase in glucose production is coming from the liver, but clearly there can be no binding. So one has to postulate an off-target effect. We believe the off-target effect is the kidney sending a signal through the renal nerves to the liver, and that's what's increasing hepatic glucose production. Now, could there be another off-target effect that has yet to be discovered? Possible, but I think that the studies that we did where we took the kidneys out in the people with polycystic kidney disease and then only had a transplanted denervated kidney is in strong favor that it's really the renal nerves, kidney, renal nerves to the liver, and probably not another off-target effect, but I would say, is there still room for a small possibility here? I guess so, but I think it's mostly the renal nerves.
Right.
Do you know whether those renal nerves going, kidney, brain, liver, or is it a direct neural communication that's happening there?
Yeah. Another, I think, very important question. And in fact, I'm kind of surprised that someone hasn't already done this study in animal models. Now, of course, in humans, you can't go in and start clipping off nerves that are going up to the brain or to the liver. But as I said, who would have even understood that there was a renal hepatic connection in year 2024? This physiology was never suspected. So here was a medical treatment, an SGLT2 inhibitor, which led to a whole new, previously unrecognized physiology. So it seems to me that now we need to go back to an animal model where we can do different types of denervation to see what is the pathway first, if it's directly from the kidney, renal nerves to the liver, or as you said, this could be centrally mediated. So Dr. Charrington in Nashville has shown that there are very important connections coming from the brain that can regulate hepatic glucose production, peripheral glucose metabolism, beta cell function. So I think this is an avenue that really needs to be explored, but it needs to be done in non-human models.
So, Dr. DeFronzo, it occurs to me that the cessation of this treatment might be an interesting time point to look at. What happens to individuals after you withdraw the inhibitors? Does the hepatic glucose production go back to normal, or is it still high?
So we've done this in a couple of ways. One obvious question is when I start you on the, we did this with dapagliflozin. We've also done it with empagliflozin and canagliflozin. So I think it's something that's intrinsic to SGLT2 inhibitors, not to one of the class. One of the first questions we were asked and we looked at is how long does the effect persist? And we know that the effect persists for upwards of 2 to 3 months because we've done the studies. And moreover, the studies that we did now were done in the fasting state. We also showed that in response to a meal, you don't get a normal suppression of hepatic glucose production, and that effect persists for up to 2 to 3 months. And that is all published. In other studies which we have not published, I can tell you that once you stop the SGLT2 inhibitor, within 1 to 2 weeks, the effect is gone. So it seems to be something that's quick on, persists for at least several months, and when you stop the medication, dissipates within 1 to 2 weeks.
Excellent. Are there any other key points or thoughts you'd like to leave us with as we close the interview?
Well, I think the key thought is, and I say this all the time, when we started these studies with the SGLT2 inhibitors, we never thought that we were going to find, hey, this drug is a great class of drugs for lowering glucose. It promotes weight loss, protects your kidneys, and it has cardiovascular protection. And never would we have thought that we were going to discover this novel pathophysiologic connection between the kidney and the liver. What I think we have to look forward to is we need to come up with ways of blocking this paradoxical rise in hepatic glucose production. And in fact, we've shown that if you can combine, and we've done this with dapagliflozin and other SGLT2 inhibitors, if you can combine them with or the GLP-1 receptor agonist, which has big effect on the liver, you can sort of knock out this paradoxical effect. and maximize the efficacy of dropping the A1C.
Nice.
Well, with that, I think we'll close. Dr. DeFronzo, thank you so much for joining us. Thanks for sharing your expertise with us today and your recent work specifically on the impact of SGLT2 inhibitors on endogenous glucose production. I encourage the listeners to read the full article in the June issue of Diabetes.
Thank you, and glad to be with you.
Welcome to another edition of Sweet Talk. I'm Kevin Williams.
And I'm Kirk Abecker.
Today we have a special point-counterpoint segment on the regulation of beta cell KATP channels in glucose metabolism, a fundamental process in insulin secretion, as well as the development and treatment of diabetes. We'll be conducting two separate interviews today. First, we will hear a perspective from Dr. Matt Merrins and Dr. Richard Kibbey on why they believe it's time for a paradigm shift in how we understand KATP channel regulation. Kirk will be co-hosting this interview with me. Then we will speak with Dr. Guy Rutter and Dr. Ian Sweet to understand their perspective and why they believe a canonical model involving mitochondrial oxidative phosphorylation remains robust and valid. So let's get started. Joining us first are Dr. Matt Merrins from the University of Wisconsin-Madison and Dr. Dick Kibbey from Yale University. Welcome to you both, and thank you for joining us.
Thanks for having us.
Great to be here.
All right, guys, we always start out with an icebreaker question. Today's question is going to be not about science. So if you were to pursue a career outside of science, what profession that you think you may have been drawn to instead? Let's start with Matt, and then we'll let Dick answer after that.
Well, when I was a postdoc, I was running a food nonprofit, and we built a lot of hoop houses, over 30 of them in the greater Detroit area. And it involved meeting farmers and seeing where our food came from, and also meeting the restaurateurs who would take the food and eating at their restaurants. And so it was great fun, and what I did when I was not in the lab. And so I would definitely pursue something in that area.
Very cool. Dick?
Yeah, so I mean, I'm assuming that we would have talent in this other area, so which I don't. But you know, in college and before, I was a music major, and I did a lot of vocal and instrumental performance. And if I actually had talent, who knows? Maybe I would have gone in that direction, but I didn't. So I stopped.
Very good. Thanks for that, guys.
Yeah, that's awesome. Transitioning back to the science, I wonder, can you both briefly summarize some key points of why a new model of KATP channel regulation is necessary? Maybe highlighting some differences between this model involving glycolysis and pyruvate kinase versus that canonical mitochondrial oxidative phosphorylation model.
Sure. So I think before we talk about a new model for beta cell fuel sensing, it's really important to describe the existing model that's been in place by our count since around the year 1984, when the KATP channel was discovered by Cook and Hales and Ashcroft. And we knew that glucose metabolism was important. And we knew that since Machinsky and Ellersman published their landmark paper in 1968. And this is the first step of glucose metabolism in glycolysis, catalyzed by the glucokinase reaction. And they showed the pivotal importance of this enzyme as the glucose sensor for the beta cell, to find that there were human mutations in glucokinase that were gain or loss of function mutations that could either cause hyperinsulinemia and hypoglycemia, or on the flip side, diabetes. And the same was found for the KATP channel. It's an inhibitory ion channel, but there are also mutations there. And so we have a lot of strong genetic information provided by human genetics, and unfortunately disease, that tells us the importance of these two critical steps. And with regard to the KATP channel, its importance was that prior to its discovery, the beta cell field was at a loss for how the plasma membrane could sense metabolism. But then it was discovered that this ion channel was not only inhibited by ATP, but it was also activated by ADP. And so the ATP to ADP ratio rise is what closes a KATP channel to initiate insulin secretion through calcium-dependent exocytosis. So that's the foundation of the model. But perhaps if there is a fault, it was an assumption that was made that mitochondria, which produce potentially the most ATP in a beta cell, would naturally be the process that everyone would think of to produce ATP to close the KATP channel. And that is precisely the process that our new model challenges. We've instead proposed that it is glycolysis, and with a special emphasis on the last step, catalyzed by the pyruvate kinase reaction, that actually has the thermodynamic drive that's much more powerful than the mitochondria to raise the ATP-ADP ratio and close a KATP channel.
Yeah, and I'd like to just add in where I think we're most confused or most confusing is the idea that we're getting rid of a lot of really important parts of the model. We love glucokinase, we love the KTP channel and mitochondria, and even we think that oxidative phosphorylation is important, but they're attributed to different roles in the system, and we're talking about a very specific role just prior to depolarization of how the glucose is being sensed, and that's where we like to focus our energy in the model, but it's not hard for people to say, hey, what do you mean mitochondria aren't important? Of course they're important. And any mutation in a mitochondria is going to affect everything else. And I think that's where a lot of confusion has arisen.
There's another aspect of our model that's not covered by the spatial hypothesis. And it's actually this concept that oxidative phosphorylation is unable to raise the ATP-ADP ratio. And Dick, could you say a bit about that?
Sure, I'd be happy to. I think it's really important, and it's a completely different area of investigation and a completely different part of the model to the spatial model, which you just laid out, Matt. And the idea is that you have to raise the ATP to ADP ratio high enough to close the KTP channel. Well, it turns out that in beta cells, almost all the nucleotide is ATP. It's in the high millimolar range, whereas ADP is in the low micromolar range. So really, the job of metabolism is to lower ADP because you're not really changing the ATP concentration practically. Lowering that ADP is what is needed to close the KTP channel. And our argument is that the mitochondria in beta cells are actually too wimpy to be able to pull that off. They're the leakiest mitochondria you can find around. They end up generating other metabolic products, other catabolic metabolites, and redirect electrons into other pathways, making them very low yield organelles that can raise the ATP to ADP ratio high enough. And it's the free energy that goes into making phosphoenolpyruvate that actually is the drive to lower ADP to the level that the KTP channel can close. So again, the idea is that mitochondria are too weak and that pyruvate kinase is the only thing that's thermodynamically disposed in the beta cell to be able to modulate the KTP channel.
Great. So after that wonderful overview, I wonder if maybe we can now go to the key experiments that have convinced you that a new model is necessary. So Dick, why don't we start with you this time?
Yeah, so I think one of the key experiments was really understanding what the role of mitochondria is in the whole glucose sensing apparatus. And as Matt mentioned, because it could make a lot of ATP, it seemed like it was the place to start. But you have to ask why beta cell mitochondria would be different than every other mitochondria that's out there, meaning that instead of being driven by the concentration of ADP in the cell, it's driven by substrate. And so that has created a lot of hand waving and wriggling to try to explain this, but there really hadn't been a good explanation. So I think one of the key experiments that really got us in the direction that we're going was the fact that we could show that beta cell mitochondria are just as ADP driven as any other cell around. and you can gently uncouple them or strongly uncouple them, and they can rise up to that metabolic challenge, suggesting that they're bathed in all the substrate that they need, that there needs to be some other way that they're connected to the signaling process. Then for me, the key observation was that there's a human disease that we couldn't explain. And it was a mitochondrial disease, and its name is hyperinsulinemia hyperammonemia syndrome. And it was really fundamentally characterized by Charlie Stanley and his group. And what this indicated was that anaplerosis, which is bringing in carbon into the mitochondria, which is not oxidative, actually takes away from oxidation, that anaplerosis was really important. And so that was the key finding that led to a pathway where we decided that oxidative phosphorylation wasn't the key ATP generator for the closure, it was pyruvate kinase.
Great. Now, Matt, building on that, maybe you can hint at what's needed, what's next to better establish or define this new model.
So, a recent experiment that's been very powerful is the observation that pyruvate kinase is present at the KATP channel. And this experiment builds on a lot of biochemical data from other groups, particularly in the cardiac field, but also using beta cell lines that show that if you can actually have a protein-protein interaction between KATP channels and pyruvate kinase, as well as other glycolytic enzymes. And so we took that experiment a step further because once you know there's an association, what you really need to know is, does the enzymatic reaction functionally regulate the KATP channel? And so what we did is we pulled off small patches of beta cell plasma membrane from human beta cells or mouse beta cells, where we could monitor the activity of the KATP channel. And we applied the substrates for pyruvate kinase and showed that it could be regulated. Now, that is satisfying in a way, but that's not an intact cell experiment. And building on that, we moved to the intact cell. And this is my favorite experiment of the whole puzzle. And it builds on what Dick talked about, this human disease. There are these mutations in glutamate dehydrogenase, which cause this hyperinsulinemic hyperanemia. These mutations take advantage of amino acid metabolism rather than glucose metabolism. And we've known for a long time that mitochondrial fuels can close KATP channels to initiate insulin secretion. Fuels like amino acids, like glutamine and leucine, especially in combination. And so we knew from Charlie Stanley's work and also work by the Kibbey lab in the mitochondria that the mitochondria could make PEP, the substrate for pyruvate kinase. And in the intact cell system, we could then use genetics where we could show that the way that these mitochondrial fuels act on KATP is not through oxidative phosphorylation. It's by making PEP in the mitochondria, which then works through pyruvate kinase at the plasma membrane. And so we have now the genetics to really tie this human disease to the phenotype of the beta cell in this new model. But I think that there is one aspect of the model that, in particular, that we've not demonstrated. We haven't shown that the location of pyruvate kinase at the KATP channel is really critical for its function. The null hypothesis, of course, according to the canonical model, is that pyruvate kinase could be anywhere in the cell, and it doesn't have to be located at the KATP channel. And so that's a key point and an unresolved point that we need to tackle.
That's great. I really love the potential clinical implications, the relation to human disease that you just brought up in your model. Matt and Dick, thank you very much for sharing your expertise with us today and your discussion on a novel outlook for the role of beta cell KATP channels in insulin secretion. I encourage the listeners to read the full article titled Glucose Regulation of Beta Cell KATP Channels. It is time for a new model. It's in the June issue of Diabetes or online at diabetesjournals.org. Welcome to part 2 of Sweet Talk. We're continuing our discussion on the role of mitochondrial oxidative phosphorylation versus glycolysis and pyruvate kinase in the regulation of KATP channels in beta cells. I'm Kevin Williams. Joining us for part 2 are Dr. Guy Rutter from the University of Montreal and Dr. Ian Sweet from the University of Washington. Welcome to you both, and thank you for joining us.
Glad to be here.
Thank you for having us.
We'd like to start these things off with a little bit of an icebreaker question. The question I have for you is not related to science. It's if you had not pursued a career in science, what profession do you think you might have been drawn to instead? Let's start with Ian, and then we'll move on to Guy.
Well, I'm not sure what this will do to my reputation, but I started off as a jazz musician and
Oh, nice.
pretty passionate and played very seriously, went to music school. That turned out to be something I'm glad that I let go and went into science later in my college career. But I certainly enjoyed that period, and I still play a lot. So,
I'm sorry, did you say what instrument you played?
oh, I play piano and actually vibraphone, which is
Oh,
a visual instrument that people usually enjoyed watching as well as listening to. So,
very cool. And Guy?
That was a scary question. I was mentally going through a list of things I could tell you, and but thanks, Ian, for reminding me. And this is a complete copy, but I also considered being a musician. I took that pretty seriously. I did all the exams and piano, and I played trombone as well. But I always sensed that there were people
Wow.
who were slightly better than me as musicians, whereas I could find fewer that were better at science.
Perfect. We had a discussion earlier with Dr. Merrins and Kibbey, and I'm wondering if you both could briefly summarize or outline some key points of the canonical model of the KATP channel regulation. How do you view this model differently than how Dr. Merrins and Dr. Kibbey view the regulation of KATP channels through the glycolysis and pyruvate kinase?
Well, if I may, I'll start. The so-called canonical model is something that has evolved over the past more than 40 years after the demonstration by Ashcroft and Hales and Cook of the important role of ATP-sensitive potassium channels in beta cells. And on this, I think we're all agreed. There's a massive genetic and pharmacological and other evidence to say that those channels are critical. We're also agreed that glucokinase, which is the flux generating or rate limiting step in the metabolism of glucose, is also critical. Again, there's a massive genetic evidence for that. So the canonical model proposes rather simply that glucose carbon flows through the glycolytic pathway and through the citrate cycle, activating the respiratory chain, which is limited by the supply of pyruvate and low glucose concentrations, and that activation enhances the production of ATP. You get an increase in the ATP to ADP ratio, largely driven by production of ATP by the respiratory chain, although some ATP is produced by glycolysis, and that globally increases the ATP to ADP ratio in the cytosol, and it's that which is seen by the ATP-sensitive potassium channels. And then their closure results in membrane depolarization and calcium influx and the triggering of insulin secretion. So it's parsimonious and it's a simple model for which there's considerable evidence. The first and very simple piece of evidence is that if you poison mitochondrial oxidative metabolism, either with inhibitors of the respiratory chain or you uncouple mitochondria with what are called protonophores, that causes a massive, essentially instantaneous drop in ATP, the opening of the channel and the cessation of insulin secretion. You can do that with other inhibitors. Secondly, almost any manipulation you do genetically to mice, which affects the respiratory chain or mitochondrial oxidative metabolism, will result in an impairment of insulin secretion and or eventually hyperglycemia or diabetes in those animal models. And there are models in human, not models in human, but there are examples of human genetic diseases, monogenic diseases, where mitochondrial respiratory chain synthesis is impaired, and these are associated with diabetes. So there's a really sort of a mountain of evidence which supports the model. At that point, perhaps I should stop and invite Ian to pick up the baton.
The 2 points I will make will involve how the consensus model or canonical model can fit essentially all the fuel secreted drugs that we know. And I'm going to make that point, and also the quantitation between the usage of ATP and generation by OXPHOS and insulin secretion. So two things. The first is, clearly we want a model to be tested, and we know about 5 fuel secretagogues: glucose, KIC, ketoisocaprate, leucine, and glyceraldehyde. And so, 10 or 15 years ago, I went through all these, as other people have done, and you get a beautiful quantitative relationship between OXPHOS, generation of ATP, and insulin secretion. So that's first and foremost what should be asked of a model, not just in trying to support it, but in trying to find cases where it isn't supported. And so this is something that we would obviously need to see from the PK model, is that the PEP and PK can quantitatively relate to insulin secretion. So that's hasn't been done in the way that we did for glucokinase when I worked in Dr. Machinsky's lab. That he was always stressing need for quantitation. It's not enough to just knock something out and saying it doesn't work or that it goes up. You really have to show that they're related in a unifying way, that whatever fuel you give, whether it enters into glycolysis, TCA cycle, or directly in the electron transport chain, that the relationship between, in our case, the ATP that comes from OXPHOS will quantitatively relate to the insulin secretion. And so these things haven't even been started for the new model. So I guess just to end that, I would say that we don't have any data that would cause us to reject as Guy pointed out, the model that's been worked on for 30 years in many different configurations, and one that's very new and hasn't generated this kind of quantitative relationships.
Perfect. Dr. Merrins and Kibbey also made a point to discuss the bioenergetics of their model. The bioenergetics argument stated that oxidative phosphorylation may not be thermodynamically capable of lowering ADP levels sufficiently to close the KATP channels and changing that ATP to ADP ratio. Dr. Merrins and Kibbey suggested that the role of pyruvate kinase in lowering ADP was crucial for this. I was wondering if you could offer any thoughts or insights on this part of the debate.
Yeah, it's a very interesting perspective, and it's a very interesting speculation, but the evidence provided is not quite as strong as the claims. First of all, it's clear that mitochondria in beta cells, as in all other cell types, when they're supplied with sufficient fuel, are well coupled, and you can demonstrate this using suitable inhibitors. And as we all learned first year at university, mitochondria are likely to produce as much as 35 ATP per glucose oxidized. Even if they were a little bit uncoupled, they would produce as much as 14. The pyruvate kinase reaction produces 2. So I would invite listeners to go away and think about that for a moment. The Kibbey and Merrins model has kind of evolved. A couple of years ago, their proposal, which was interesting and may not be completely wrong, was that at high ATP, at high enough ATP levels to close ATP-sensitive potassium channels, ADP would be limiting for mitochondrial oxidation by the respiratory chain. Although there's not really direct evidence for that, it's a reasonable thing to hypothesize. However, the affinity of pyruvate kinase and mitochondrial ATP as synthetic machinery is just about the same. Actually, the affinity for ADP is slightly, or the half maximal concentration required is actually a little bit lower for mitochondrial oxidative phosphorylation. So it's difficult to argue that as a reason. More recently, the Merrins and Kibbey model seems to have evolved. Rather than claiming that mitochondria were too efficient and there was too much ATP, which would mean a freezing of ATP synthesis, this has evolved to the one which you just mentioned, whereby the claim is that mitochondria are too inefficient and they cannot make enough ATP. This, as I just explained, is a little bit difficult for us to resolve, given the bioenergetics, which will say that there are something like an order of magnitude more ATP produced by mitochondria. So the hypothesis around microdomains is an interesting one. This becomes important if you can provide really strong evidence that indeed pyruvate kinase is in very tight association with close proximity to the channels themselves. And this seems to be the case in other tissues such as the heart. This is yet to be demonstrated fully or compellingly in beta cell at endogenous levels of the proteins themselves. And we think that a demonstration of that association is absolutely critical. Moreover, one would need, having demonstrated it, to perturb it, to dislocalize, to relocalize the pyruvate kinase enzyme away from the channels, and then explore what effect that delocalization has on ATP-sensitive potassium channel closure and insulin secretion. That is, for us, a critical experiment. The evidence that has been obtained using a patch clamp electrophysiology and supplying phosphenylpyruvate, which it is argued will locally allow pyruvate kinase glued to the channels to produce ATP, is something which hasn't unfortunately been replicated by another lab that has tried to replicate those data, even though they can replicate the data from the heart. So that for us is an important limitation. In contrast, the evidence we have for the canonical model has been replicated in multiple labs. Naturally, this has been around for almost 40 years, so that's unsurprising. A final point I'd add, just to step back a moment, is if we look at the metabolic configuration of the beta cell, it's quite a remarkable machine in terms of the production of CO2 and water from glucose. About 90% of glucose is converted to carbon dioxide and water. And if you want to find any other system where that is the case, you have to look at insect flight muscle, which produces colossal amounts of ATP to drive muscle contraction and flight in the fly. So although that's a teleological argument, it does tell us that the beta cell is a remarkably efficient machine for making ATP. So to put it in these terms, why would you require to have a local domain of ATP? So next to the channel, because mitochondria are in pretty good shape.
I could sort of give one sort of different perspective on it, which I think, was hammered into me by my mentor, Dr. Machinsky, in that he thought like an engineer. How could this system work teleologically, right? That like if you had in a house, you have a thermostat and a heater. When the temperature gets cold, then the heater goes on. It's very simple. And glucokinase, as a hypothesis, as a control mechanism, plays that simple role, that it controls the flux and then of glucose into the system. And then you can, if you propose that from then on, it's like a pipe so that you generate a given amount of ATP, then you have a system that is controlled by the glucose sensitivity of glucokinase. And that is allowed to, that will change in the physiologic changes of glucose. So if you then relegate control of the system by an internal mechanism like pyruvate kinase, which is many steps away from glucokinase, how do you retain that physiologic glucose sensitivity? And it seems just anti-intuitive that instead of having a system that's really based on control by extracellular signals rather than intracellular, you're changing the beta cell from something that will be dependent on its own energetic needs, depending on how much PEP is coming from the TCA cycle versus how much is coming from glycolysis. Now you have a very complicated system. And so this really doesn't even kind of make sense that this is the way it would work. And as Guy has pointed out, there's really not data that would take away from the very logical and sensible model where it simply produces the amount of ATP and signals that the glucokinase is providing the control for as the initial step.
I think if you allow me to pick up on that. I mean, there are critical measurements which would support the model of Doctors Merrins and Kibbey, which are still missing. What one would need are many more measurements of phosphenylpyruvate, of PEP, under conditions where it is argued that that is the critical regulator of the channel. One of the points that I would make is that after the initial response to a step increase in glucose of the beta cell, where I think the evidence is overwhelming that increase in oxygen consumption, ATP synthesis, is the result of increased pyruvate, you have a more subtle and complicated set of events which occur when oscillations begin to occur. And these oscillations I'm talking about oscillations in calcium and in exocytosis. These may well involve limitation of ADP supply to mitochondria, but this is resupplied when ATP is broken down because you start consuming that molecule when calcium flows back into the cell. One of the problems with invoking oscillatory changes between PEP supply and oxidative metabolism, which has been one of the models that was proposed in their review in Cell Metabolism a couple of years ago, is that by having phosphenylpyruvate producing the ATP equivalents, if you will, for closure of ATP channels, you'd be doing that exactly at the time when, in fact, you'd rather want the channels to reopen in order to have an oscillatory pattern of firing. So that doesn't seem intuitively helpful in terms of providing a model for oscillatory changes. In fact, it would be more helpful that the channels would reopen because ATP would fall a little bit in the cell as a whole.
Thank you, Guy and Ian, for joining us today. I encourage our listeners to read the full article titled Glucose Regulation of Beta Cell KATP Channels is a New Model Needed in the June issue of the journal, or online at diabetesjournals.org.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope you are enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournals.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you next month.